transcription. It involves not only ribosomes. By the way, ribosomes don't do it. They provide the location for it. What does it are a whole bunch of enzymes and transfer RNA molecules and things like that that can bring in right amino acids and read the message. This message that we read for every 3 base pairs we see here, that's called a codon, we will specify and put in a particular specific amino acid. So these adapter molecules, the translators if you will, that can actually read the nucleotide message, convert it to an amino acid message, is a transfer RNA molecule. Okay. And so they're vital in all of that, and they have to be charged with the right amino acids to bring in, to sit down. It's a very complicated process. And obviously if errors occur anywhere in here, you're going to, if an error occurred here, you've changed the structure of the RNA. If you change the structure of the RNA, you're going to change the structure of the polypeptide. Sometimes it doesn't matter. Most of the time it does. So it really kind of depends. If you have an error in translation, again, you're going to put in a wrong amino acid. Sometimes not a big deal if they're chemically similar to each other. Sometimes a huge deal, meaning that you've heard of sickle cell anemia. Okay, just to give you, because this is a common thing a lot of people have heard about. The only difference between normal hemoglobin and the hemoglobin, the sickled hemoglobin, is 1 amino acid out of about 400. So that's how simple some of these changes can be that result in drastic changes, right? A structure function kind of thing. Because we know what happens here is once you have this polypeptide, this string of amino acids here, that has to fold. And the folding, right, goes through, they're on the board there, I knew there was a slide somewhere of it, through several different layers, primary, secondary, at least tertiary, and sometimes even quaternary levels of structure, right? Folding, 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 interacting, right? Until you ultimately produce a protein. And that protein is something that's functional. That's all that means. It can do something. And what can it do? Well, it depends on what it is. It could be a fibrous protein that is just being used for like collagen or the cytoskeleton. It could be a globular protein that functions as an enzyme and has an active site that binds to a substrate and catalyzes a reaction. It could be a transporter that'll bind to some kind of, we call them ligand out there, and move it across the cell membrane. It could be a hormone that signals like insulin that triggers the uptake of glucose by cells in the body. So it really depends on what it is, but if you have any little error anywhere along the way here, there can be issues. It's kind of interesting.